Mhm. Hi, everyone. Welcome to the fifth Spatial Omics seminar series. Today, we are very happy to have Dr. Joseph Beechem from NanoString Technologies. And briefly, Dr. Joseph Beechem holds the position of Chief Science Officer, SVP Research Development at NanoString Technologies. And he previously held the position of Chief Scientific Officer at Invitrogen Corporation, CTO at Life Technologies Corporation, and Biosciences Director at Molecular Probes. And also he was in academia actually for some time as an associate professor at Vanderbilt University. And today then we are excited to hear more about Spatial Molecular Imager, a complementary technology that Dr. Beechem presented a few months ago. And without further ado, I'd like to turn to Dr. Beechem, please.
Hey, thanks, Ahmet, and thanks, Rong. And like I said, I love this series. So for me, this is a real pleasure to be able to come here today and talk about, you know, if I can get this thing running, let's see what I got. Talk about this brand new spatial molecular imager, and you know, toss stuff into the chat there. I'll try to watch, you know, how it is when you're presenting in presentation mode. Hard for me to see all the things, but you know, stick the things in chat there. I'll take a look at it as I go along here. You know, I think when I talked a few months ago, I really emphasized the panel on the left here, the multicellular whole transcriptome geomics digital spatial profiler. That was a technology we launched at AACR meeting in 2019. Now there's a couple of hundred of them out there into the real world. And, you know, it's really good at what I call doing whole transcriptional profiling on sort of the functional units of organs. If you actually look at the functional units of organs, they actually range in size. A single cell doesn't do a function in an organ, but things that are on the order from about 100 microns up to about 2 millimeters actually do accomplish functions. Things like the glomerulus of a kidney is about 100 microns. Islets of Langerhans and a pancreas is a couple of 100 microns. So these higher order single cells all assemble to do function. And that's really what Geomics DSP was designed to probe. But at the same time, we were working on a subcellular and single cellular high-plex technology called the Spatial Molecular Imager that I'm going to emphasize today, where you actually can look at the private lives of the cells as they organize into these aggregate functions. So I'm going to do one little vignette on the Geomics DSP, just because I think it's of very much general interest, and then I'm going to spend the majority of the time talking about the spatial molecular imager. So this just sort of highlights the different spatial scales I think it's important to be able to probe. And I think being seamless between these spatial scales are really important. The other thing I'll mention is that all the technologies I'm talking about today are multi-omic. So we're doing both RNA layers of information and the protein layers of information. So I think you need a couple of spatial domains, you need high-plex, and I believe both the RNA layer and the protein layers are very, very important to get them all to work together. And if you can do it non-destructively, that's even the best. So most of the technologies, in fact, all the technologies I'll be talking to you today actually doesn't destroy the tissue you're measuring on. So you can still do things afterwards. So let me do one little vignette on DSP, the Geomics DSP, just because it's data I collected since the last time I talked here. And I really think it showcases the role of whole transcriptomes and what we can get out of them and how you reference against known databases. I'm not going to introduce DSP, just to others say that the data I'm going to be showing you is the work on the RNA reagents down there. These are fish probes. We're looking at about 18,500 fish probes in space. All those fish probes have little photocleavable linkers. So when I go into an organ and want to measure a whole transcriptome around a particular region, the mirrors that you see on your right will adapt to a single cell resolution, the shape of that unit inside that organ that I want to measure. You'll shine light on just that part of the organ. And then the photocleavable indices come off. You sequence those. We sequence them on a Illumina sequencer, take high resolution imaging at the same time, and then bring all that back together. So that's how I'm going to show you a whole transcriptomes on these units. And that's how it's being collected. So I just wanted to do a little vignette on this example. Take something like a kidney, has all this interesting macroscopic structure like that. You see the overall whole kidney. It's actually a medullary region and a cortical region. But look on the right there, the business end is a glomerulus, and that's about 100 microns of cells that work together. That is really one of the key business ends. But then that empties into what's called a proximal convoluted tubule. That goes into a distal tubule, and then it empties. And I'm going to do whole transcriptomes as I walk my way from the business end of that organ, that little glomerulus. I'm going to do whole transcriptomes, and I'm going to move into the proximal tubule, do whole transcriptomes, move into the loop of Henle, do whole transcriptomes, move into the distal convoluted tubule, do whole transcriptomes, and just watch the biology move as you move through an organ like this. So here is sort of schematically, now with some real data. You see the little cartoon thing on the left. Here is the full-blown, you see the whole red, that's the cortical area of the kidney, and then the green is the medullary region of that. I'm going to go into one little business unit, one area around the glomerulus. You can see that blown up there. There's a couple of million of those in a typical human organ, to give you an idea how many of those little business units are in there functioning. So there's the glomerulus. I'm going to have mirrors that would just map to that exact shape of that glomerulus. And then you can see that little green arrow. I'm going to get a whole transcriptome there. And then I'm moving farther down, say, proximal collecting duct, that upper right green one. I'll collect whole transcriptomes on that, and I'm going to work my way down through the path of the biological function of a kidney. Now, when you do that, and I didn't know what this data was really going to look like, it's because the first kind of data like this that's ever been taken. And what you see is like, I've been amazed at this, to be honest with you, is that you can just plot the heat map of all the pathways in your whole transcriptome, okay? And then look at the region of that little heat map, says proximal convoluted tubules there, okay? And then look at your right, you're just plotting out. I'm just taking the highest scoring pathways that I'm getting when I'm in that PCT, that proximal, and then you see things like sodium phosphate co-transporters, sodium ascorbate transporters, so on and so forth. And if you actually go back through all the textbooks on kidney physiology, these are exactly all the transporters that are supposed to be in that little proximal convoluted tubule region. And then, okay, so then what happens when I move my transcripts farther downstream? And now I'm in the distal convoluted tubule, the little thing called CD there. And what happens? What shows up there? And let me show you that. If I can get my screen back. There you go. And you can see as you go to the length of those tubules, you actually come down into metal transporters, choline, and at the very end of the distal convoluted tubule, one of the last things that's transported is the ammonium transport. So you can see it's sort of like one experiment on a healthy organ like this. I'm just walking myself spatially with whole transcriptomes through the biology of how that organ functions. And I'm basically reading off, as far as I can tell, about 50 years worth of kidney physiology recapitulated. Those are all individual studies, crazy, dissecting organs and ion channel recordings and all these other things. You're getting about 50 years of kidney physiology recapitulated, a single set of whole transcriptomes on this particular tissue. And then I said, well, that's great. Let's say we recapitulated that. You also see interesting new little things. I'm going to highlight just one little thing. See that one little thing with the ABC transporters? All of a sudden showing hot there, those two little reds. I'm not sure if you can see my little arrow or not. very likely this person was actually on some, when you take drugs and you're on certain painkillers and things like that, those ABC transporters come up. I think that's actually what we're actually seeing there. So you actually see a whole lot of extra interesting things at the same time. Now the question is, how do I know what I'm measuring is right? And what you find is, I was really interested in Emma Lundberg's talk she gave a couple of weeks ago here. And the Human Protein Atlas is what I've found to be is an atlas I can go to, because there is no other information. You can't go in there and find whole transcriptomes of a glomerulus and a kidney. You can't just look it up. This really hasn't been done. So I said, but I can go into the HPA. I can go to the Human Protein Atlas and find all these proteins that subcellularly localized in these regions of the kidney. And I can plot that along the y-axis here. And I can take my whole transcriptome data and plot it along the x-axis. Yeah, what do you find out? Lo and behold, all the heat maps from the whole transcriptome as I move through space map exactly with the protein subcellular locations as determined in the Human Protein Atlas. I was really amazed by this. I think it's probably the first time. I sent Emma a note last night. I believe it's the first time HPA has ever been cross-validated at the RNA level in this kind of manner, at the whole transcriptome level. But maybe she'll be able to tell me whether that's true or false tomorrow or something. But then, okay, that's it at the global level. Let me give you an idea what it looks like individually on some of these individual proteins. I'm only going to show you 2 little cases of that. So you can see over here, the data on the left is the Human Protein Atlas image of something called HPA protein CALB1, whatever the heck that is. But it's a protein that only shows up in the HPA around the little distal convoluted tubules. You can see the little dart there. And then what do I get when I do the whole transcriptome and I'm just taking the CALB1 message? Where does it show up? You can see the only place that message shows up is in the distal tubules. All the other parts of that little train that I was walking doing whole transcriptomes are nothing. You hit that particular region, it pops up there. Exactly recapitulating the information in the HPA for both the target and the location. Here's another example here. This is some other protein, SCLC3, whatever this thing, solute carrier family 34 member 1, whatever the heck that thing is. And you can see that it showed up in the HPA only in the proximal convoluted tubules. So I go over to the whole transcriptome stuff that we're doing. I just plot out that particular gene. You can see it's nowhere until I hit the proximal tubules, and then it pops up. So, I think this was kind of a fun one for me. I didn't really know what these whole transcriptomes would look like and how you can just follow the biology. Like I say, you recapitulate about 50 years of kidney physiology in one experiment, essentially. And then you can cross-validate it against the protein maps in HPA. And, I just amazed at that, because a lot of people say, well, then how's the RNA correlate with the protein and all that things? And what I really find is that probably 80% of the time where that correlation they say it doesn't exist. And there's real reasons. There's a bunch of reasons why it doesn't exist. But a lot of stuff in the literature is just because the methodology they use to compare those 2 things is so bad and so weak. I think it's almost meaningless. You can see here on this scale, great correlation. Okay, so let's move out of the world of sort of the functional macroscopic way these organs function. And then let's just go into the private lives and how do we develop technology to really study the private lives of the cells that aggregate together and make this happen. So this gives you an idea of the kind of images that are coming out of Spatial Molecular Imager. I think in one talk I called this thing the Hubble Telescope of Spatial Biology, and sort of caught on in some respect. Each one of those little dots you're seeing, we're looking at a melanoma tissue here. Each one of those little dots is an individually counted single RNA molecule in an FFPE tissue section. We're doing about 1,013 different RNA classes at a time here. When I'm looking at this thing, when we launch this next year, you'll be capable of doing about 500,000 cells like this generating about 1 billion transcripts off a single tissue slice. So that's the scale we're working at. 500,000 cells, 1 billion transcripts at a time. Okay, when we're developing this, actually, it started as a different kind of project. It started as a project where I was inventing and developing ultra small cyclic optical barcodes that do sequencing. And it turned out we came up and we developed a 20 nanometer sized a barcode, optical barcode that will change colors as I put different hybridization reagents on there. And it's 4 different colors. So you encode information like that, 4 colors, and then how many bits of information is how many cycle events you want to have and all the light dark patterns. We developed that early in 2016, and we were going to be developing a clinical grade sequencer with that. We actually published on some infectious disease detection with that, and in Nature Medicine. I think that was a paper we did, forget what year that was, 2019, I believe. But as we're doing that, I was thinking it's a total hybridization-based sequencing reagent. And then if I look at what I'm doing with that, this is sort of what I was doing with that reagent originally. Look at the left-hand side of your screen here. You know, I was taking Mother Nature's FFPE, yanking the RNA out of it, putting it down onto an ordered array, and then using a hybridization sequencing reagent and sequencing all these things in parallel. And you really think about it, it's like, why go to the work of pulling it out of the tissue? Wouldn't you rather just take Mother Nature's ordered array, which is the tissue itself, leave the RNA where it is, and let's bring that hybridization unit into the tissue. And then now we're quantitating the RNA and high-plex. Now we know where it is. So that's what we did. Starting about 2.5 years ago or so, 3 years ago, we started adapting this to do Nc2 detection of RNA. And I'll show you how it works now. It works just like all the other NanoString technologies. We built all these chemistries to work on FFPE. It's always the hardest one. My figure is just do the chemistry right, do the hardest one, and the rest of them fall out. So that's what we did here. It's the same thing we did with geomics. So you just take a standard FFPE slide, you do a standard prep on it. You could even do it on an automated like a bond tissue processor if you want, through the Nc2 hybridization of these reagents. And I'll show you how the little Nc2 tagging thing works. Goes into a flow cell, put it into the instrument, you close it up. Actually, it's what the outside of the Spatial Molecular Imager looks like. There's a kit that goes in, you run your reagents, you push your button, and then what you get out are these things to the right. So the information content, obviously all your tissues, you got your 1000-plex RNAs, so you can do your UMAP clustering. All the UMAP cluster all have X, Y, and Z coordinates to them. So you take all your clusters and you put them back into space. That's what the actual raw data looks like. And then you can do a whole series of different types of analyses at the single cell level. And I'll show you what some of that looks like. But it's a single tabletop box that gets you from A to Z here. I'm going to bring you under the hood on how the chemistry works a little bit. So how to be able to, it's fun to be able to talk to a technology group like this. So anyway, so here goes. If you know how geomics works, you got the hybridization, Nc2 hybridization. Then there's a little tail that's got the sequencing barcode on it connected with a photocleavable linker. So we do our hybridization, and then we put the light on and a little tail comes off, and then we sequence Nc2, if you will. But on here, we wanted to bring the imaging reagents in. So we do our hybridization. There's a little tail on what we're hybridizing on. You can sort of see it over here on the far left. So there's Mother Nature in red, the little RNA there. Here's our business end hybridization. There's a tail that sticks off of that hybridization complex. And what I'm going to have, my sequencing barcodes, that little 20 nanometer little sequencing barcode rides on the back of a hybridization reagent that goes cycling in. And if it hits the right pair, and the encoding for what RNA I'm on is in the color in that tail. So then in this case here, if I'm making a readout of 1,013 different things, I actually encode this information into 4 colors, 16 cycles, so 64 bits of information. So I cycle a reagent in. If there's one that has a landing pad for, say, the blue reporter group, and it lands and sticks, that means it's a hybridization event. That means that records as a blue that you see down here on your left-hand image. These are all single molecule images here. And you cycle that reagent off. The whole image goes dark, and you bring your new reporters in. They have a different little landing pad on the back there. And then in this particular case, I'm sort of saying that a little yellow reporter hit on the second cycle. So I would have measured down blue spots turning into yellow spots, and I'll blow up this image on the next slide so you can see it a little better. But you encode this information where all these little single molecule RNAs scattered over this entire tissue section are changing colors. And that color pattern identifies all the little transcripts that you're trying to measure. You can just encode, there's a sort of a schematic of the information. It's like a 4-bit. It's each bit has got 4 features associated with it, if you will. And you cycle through and you generate your full resolved image. So when you're looking at the raw data, that's sort of where it's coming from. If I blow those things up, you can see them a little better. Here's the little blue. There's a single RNA molecule there with one of our 20 nanometer sequencing barcodes on it. Here's another one. We cycle out to yellow, and there you can see a yellow one that's positive on this guy. We're cycling later on back to blue. They turn blue again, and you're doing this over your entire tissue section. That's how you encode it.
Can I have a quick question?
Yes, absolutely.
What happens when 2 RNA molecules are really close to each other?
Yep, it's an important thing. And so one of the key things is when you're doing imaging reagents, that's why we do no amplification on our imaging reagents, okay? So they're the smallest possible bright spots. So that's one reason that they're packed, roughly 30 dyes get packed into a 20 nanometer barcode. So making them small is good. That means the crowding happens later. You can crowd them in there a whole bunch, and I'll show you how many you can see at a time. But then the other way to control the crowding term is that I can control the series of sequences. So when I'm doing 4 colors at 16 cycles, some of these cycles are dark. So I am actually turning a whole bunch of things on and off. So you can actually have a couple of RNAs show up inside a single diffraction limited spot. But if it obeys a different on-off dark light cycle rule, you can actually resolve both of them, actually, even though they're in the same diffraction limited area, because I'm spreading the information out into different cycles. It doesn't mean eventually you will run into a crowded thing. It's pretty easy to see when you have multiple things that aren't even resolvable at that cycling, but you can actually do a lot. And it surprised me how much you can do. But anyway, it's a long-winded answer to your crowding question. Is that helpful?
Yeah, thanks.
Okay, so let's take a look at this thing and how it actually goes. I think kind of interesting to keep your tabs on how big is these data sets. So when I'm in there doing this cycling and doing these images over all these tissues here, and now you know these details of these little colors going on and off and so on and so forth. We generate, the raw data is about 10 terabytes that's coming out. That's a pretty good chunk of data. At 10 terabytes, actually luckily never actually has to move out of the SMI. It sits there. All these things then get decoded. They get turned into actual digital genes or proteins. And all those digital genes and proteins have an X, Y, and Z coordinate that you can see over here. And once you've sort of digitized it at that level, you're down to about 1 terabyte of information, which is more manageable than 10. And you go one step farther, and then you actually go in and you start segmenting the cells on the right-hand side, where I actually know where one cell stops and another one starts. And now I know where all my RNAs are in these individual cells. Then you end up with a data set that's actually all the way down to about 1 terabyte. It actually has the gene identity, color codes, XYZ coordinates digitally now. The location of where that RNA is it in cell number or whatever, 1938 or 1205 or 304,003. They're all indexed that way, where they're at, cytoplasm. Your actual data set actually gets down to a pretty manageable thing that's down to about 1/10 of a tera. Okay, I see another one, a good question in there that I'll get to. 1/10 of a tera. But anyway, that's sort of the cycle of data through the system. Okay, this is sort of what it looks like now. I'm back into an FFPE melanoma sample here. You got the XYZ coordinates of everything. So I'm down into about a 1/10 of a tera database now with this study. And then you do exactly that thing I was talking about. You have all that information, you map it, you take the XYZ coordinates of everything, you reconstruct the tissue, and then you have all these extra genes that you can actually start looking at differential genes as I go through my mapping clusters. Like this is a differential heat map for a cluster. Let me find one that I can read with my glasses here. Hey, let's take a look at cluster. I don't know. Yeah, cluster 5 is the first one in here. Oh, okay, cluster 5 right over there. This little green cluster of cells got a whole bunch of markers here that are hot for in that particular cell cluster, a UMAP cluster, if you will. And then you can just walk your way through the clusters, reading off the gene specific with those particular clusters. Now, let me tell you a little bit about the biology that comes out of here. This is our first panel we've been working with. It's a 1013 plex panel. And it was really designed to answer these real simple questions like, who am I? Where am I located? What am I doing in this location? What's my cell state and function? You can see roughly how many genes it takes to do those things. Cell typing and mapping actually only takes a couple of hundred genes. You can actually map a lot of cell types with that. Cell state and function, we devote about 270 genes for doing that. Cell-cell interaction, we're really interested on how the private lives of these cells as they talk to each other. So I really loaded up on all the cell-cell interaction and how the different receptor systems talk to each other and all of that. And then cells sometimes are affected by signals from far away, hormonal activities and things. So we wanted to have things like that. We sort of view this 1000-plex as sort of like table stakes, if you will. If you really want to look at a single cell, you want to understand these kinds of questions. It takes about 1,000 genes to do that. A half decent job of it. Okay, this is what it looks like when you're doing this, and we've got a lot of different types of FFPE samples run through the SMI at this stage. And I'm just showing you a handful of these. We do a lot of disease-based research here at NanoString, so that's why most of these things look like different types of cancers and things: melanoma, non-small cell lung cancer, renal carcinoma, colorectal. We also look at things like tonsil, lymph nodes, those types of things. But in every case, you can do this high-plex, high-efficiency cell identification and then start looking at functional differences. I think, if you're a single cell person, one of the key things is that there's really no dropouts. And when you're doing this kind of work in a spatially resolved manner, it's not like single cells where you get these snippets of these high expressing genes, and then you may pile 10,000 cells together and get an histogram. You are deep in every cell. There basically isn't any dark cells here. And you're measuring, you're going deep in the gene content of every single cell and no dropouts. Now, if you want to, you can actually put a bunch of tissues on a single SMI flow cell, and that really allows you to get more samples going at a time. This is one we were running the other day. Again, these are non-small cell lung cancer tissue arrays. What are we looking at? 1, 2, 3, 4, 5, 4, 8, 10, 12, 15 different tissue slices at a time, doing all this massive cell identification on all these particular types of tissues simultaneously. That definitely helps your throughput. Let me see, I'm gonna maybe answer a question or two here before I launch, before I get too far behind them. This question on SMI technology, similar to SeqFish Plus and Long Kai's lab. Yeah, so one of the things that one of the major differences between us and a SeqFish Plus or a MERFISH approach is that again, these are things we optimize for FFP kinds of studies, where an individual RNA tends to be fragmented, so you don't have a lot of space. I can't tile a whole bunch of things along a large region because in an FFP, those lesions aren't typically connected to each other. So we really designed it. We can actually go in there and do our high-plex profiling, and all I need is about 100 bases of contiguous RNA, and I can identify it. So that's one of the things, but it's similar. We encode, all these methods are going to encode information. So in that respect, the encoding, there's a million ways to encode information. We have our own way of encoding in these really small particles. But the other thing is that we look at a very small footprint of a little small footprint of the RNA that we are required to get. Okay. There's a question that's coming up on, okay, error recovery, that's right. When you encode 64 bits of information, give you a whole bunch of ways of encoding information. I'll show you what the false positives and the error rate looks like here in just a couple of slides. Okay, and I'll show you about proteins. So those are coming up. So the next couple of questions are already in my deck. So let me get back onto the flow of this. Okay, so now we're going into real FFP tissues. And you notice the importance of segmentation here. These cells aren't little balls or whatever. It's important. So we use the protein layer to determine the actual cell outlines. So we're using same kind of hybridization sequencing chemistry, but now we're on the back of an antibody. These antibodies are ganged up on membrane proteins. So that's how I generate those little aqua lines here. So I actually know where the RNAs go at the single cell level. And now this is really sort of a full-blown version of that. I view segmentation as still slightly unsolved problem in this area. We spend a reasonable chunk of time on it, and I'm pretty happy to have gotten it to this place. So I'll just walk you through this. You can see the melanoma tissue over on the left-hand side. We're probing it with this 1000-plex. So inside that 1000-plex, but I really want to know where all those 1000-plex RNAs are in which particular cell. We also got a DAPI nuclear stain on there. So you can see now I'm going in there and generating them, blowing up the segmentation. So look at that in that lower middle. You can actually see cell outlines. You can see all their different shapes. We actually got the DAPI nucleus inside each one of those two, because these cells all don't line up like this. They're actually curving around, and it's all dependent on how the microtome chopped through this particular tissue. And that's where you can actually almost sense the planes of these cells where the microtome came through by looking at you're actually watching a nucleus rotate around in space a little bit, if you will. But, you know, we're really happy to get it to this place. We're still going to be taking it much farther, but I really like being able to use this. Is a case where the protein layer of information really is super helpful into how to pack the RNA into the right cell, if you will. Okay. And there's a question about how to hit the membranes. I mean, you know, I can't tell you all the membrane targets we hit there, but it's not exactly rocket science at that stage of the game. There's a whole bunch of integral membrane proteins that are pretty much common to almost all the tissues of the body. I think a variety of those. And are we done with our cocktail? Probably no. We'll continue to develop that. Okay, so this is what you get when start quantitating these things a little bit in terms of telling you how many RNAs are in each one of these cells. Here's a distribution over a number of cells here. you can see that sometimes we have cells where we're quantitating as many as 4,500 transcripts in a little tissue cell and maybe 10 microns on a side. So you can really, you can see a lot of transcripts in here. If you go back and look at the medium transcripts we're measuring, it's around 1,250 transcripts is our median per cell on these kind of tissues. And then on the right-hand side, I'm sort of quantitating them, the number of counts per cell. I'm using a couple of example case, high expressors, medium expressors, low expressors, and then we got these negative probability, negative probe 3, I think are labeled on here. And I'm going to start on a negative probes because it's something almost none of the high-plex RNA technology papers have in there. I don't know how they do it. They're absolutely essential, I think, for actually knowing what your real backgrounds are. So you guys probably in the RNA-seq business are familiar with these Ercc controls. These are sequences that don't exist inside human bodies on planet Earth anyway. So we actually, inside our assays, we always have these Ercc negative controls. I guess sequencing barcodes are going to combine to those things and recognize them and everything else. But it just turns out that those sets of sequences aren't in humans. It's really useful to do because then you get an idea of what your sort of real assay dependent backgrounds are. And then here they are, if I look at that thing. If you actually quantitate these Ercc controls, what you the noise on this particular system is roughly 0.1 transcript per cell. Okay, so that gives you an idea. So when I look at something, that's my baseline background noise. If I look at a low expressor, say there's something that's 1 to 2 copies per cell, typically. This is some gene H4C3, whatever that thing is. But anyway, that's its distribution. It's known to be roughly 1 to 2 copies per cell. And you can see when you're quantitating that and your noise level is 1/10 of a transcript per cell, those low expressors are easy. You almost never miss them. That's why you don't have dark cells in these kinds of assays. They all, because you're seeing them at high efficiency, even at low counts. Then as you go to medium expressors and high expressors, you can sort of see the individual count layers on there. But I was pretty happy with this kind of background and being able to see the low expressors at this kind of level. Now you say yourself that, again, like we did in geomics or whatever, we do all sorts of tricks to say, how do I know the things I'm measuring are right? Super fans, we made a ton of use of, there's a nice database of single cell, a nice deep RNA-seq database that we use a lot that was generated by the Broad. It's Cancer Cell Line Encyclopedia. It's about 1,000 cancer cell lines. They took like roughly 300,000 to 500,000 cells of those, do deep RNA-seq, and they made a database over those 1,000 cell lines. We take those exact same cell lines, we make FFP pellets of those, and then we look at our 1,000-plex assays on them, and then we make plots where we can actually put the RNA-seq from CCLE on the x-axis, and then what I do, what I'm counting, in this case here, single cell counting, and it looks like we're adding up about 3,000 cells and just plotting those things out. And then how well do they correlate? And these are the kinds of correlations that we measure. These things map with the predicted RNA-seq and all these different cell lines. And those things are all different. If you ever look at those, there's solid tumors in there, liquid tumors in there. They are all very, very distinct cell lines. So when you get correlation of your 1,000-plex over all these cell lines, over all these different types of biology, you can be pretty darn convinced that the biology you're measuring in these assays actually represents the biology that are in these cells, and also the biology that are in the various tissue types that we're measuring. Also, that's some of the kinds of validation we do. We also run RNA scope on a lot of these things and quantitate the little RNA scope dots with these higher plex assay dots and all those types of things. So let me show you, let's do something with this thing. There's a couple of things. What's it like to hunt for rare T cells in something like here, we're in an FFPE kidney cancer case, and interacting with the investigator right a couple blocks away from me here, my labs here in Seattle at the Fred Hutch. He discovered a really fascinating class of T cells. I called them bystander T cells. And then there's bystanders, and then there's ones that are active, and we're on a hunt for the active ones. I won't go through the whole poster, obviously not enough time to do that. We showed a lot of this at AGBT first week of March. But this is the flow of information now on a real sample. There's an FFPE kidney sample. There's the UMAP clustering of all the cells we're monitoring in there. And you can see various classes, highlighted plasma, B cells, so on and so forth. And now remember, like I've said before, each one of those elements of the UMAP, each data point in a UMAP has got an XYZ coordinate associated with it. So then we can actually take that UMAP and then map it into space. And I sort of left it there so you can see the FOVs, the individual FOVs along the overall macrostructure of this kidney sample here in this upper right quadrant. So these are all ID'd now, probably about greater than 95% of the cells in that tissue would have been ID'd, identified from the RNA there. And we do things like in the lower left, we're actually looking at gene expression in what I call sort of neighborhoods. Like, are there groups of cells that tend to behave together in their gene expression pattern? And do these things spatially locate? And just like you do in geomics, DSP, you find out there's incredible action in terms of changes in gene expression pattern where boundaries meet each other, where one tissue boundary meets another. You see these all over the place in tissue. And the same thing happens here at the single cell level. You get regions where two different cell types meet along a boundary, and then you get unique gene expression that's linked uniquely to that boundary. And then sometimes there's gene expression patterns that span a wide spatial domain. So we combine these sort of functional neighborhoods with the cell identification, make overlay graphs on here. And then I'm just plotting specific types of T cells now in the right quadrant, right lower quadrant, and then superposing that on these sort of functional domains where weird things happen in tissues. And yeah, you can explore these data sets. They're so information rich on so many aspects of biology, but just sort of giving you a feeling for sort of what first pass kind of things you can do. And this is the reason why all of us do spatial biology, because the information content is so dense and so rich. Okay, I want to give you another little bit of a flavor of a particular application of this, and I'm sort of excited about this one also. We're talking about CRISPR-Cas9 screening. So instead of just being passive with your tissue or passive with your cells, monitoring what they're doing, let's take and go proactive. Let's start engineering and do some engineering things like with CRISPR-Cas9. And then can we get functional readouts of cells at the same time we're engineering them? So this is what a CRISPR-Cas9 experiment looks like inside the Spatial Molecular Imager. What we're actually doing is using some of our barcodes, our sequencing barcodes, to actually in situ image the guide RNA, which guide RNA is in that particular cell at any given period of time. Sometimes you have multiple ones that will actually be in an individual cell, so that you can sort of see that over here on the right-hand side on a blow up. You take a particular cell, take that one with all the little green dots in the upper right-hand side, that would be just one guide RNA, and you can count how many are in there. And then you look right at the cell next to that green one, you see a mixture of both green and red in there. So at least probably two types of guide RNAs in that particular cell, no guide RNAs in the one cell over next to it, so on and so forth. So you end up with this map, spatial map, of the guide RNAs and the combination guide RNAs that are in each individual cell. Now you got 1,000 plex worth of genes that are coming along for the ride here. So if you look at the guide RNAs, just look at the panel right underneath there, that'd be the 1,000 plex phenotypic readout of the change in those 1,000 genes that are related to the operations, the active operations doing at the guide RNA level. Okay, So in this particular investigator, this is actually a guy named Matt Friedman up at Dana-Farber in Boston, doing this work with Matt's lab. You know, he actually, you know, he's a high throughput functional genomics person, and I'll tell you what he said about this experiment at the end, perhaps. He's interested in how antigen receptors work and his prostate cancer. So the cells you saw up there were called LNCAP cells, and he's knocking down, you can see various regulatory elements of the antigen receptor, really key receptor for prostate cancer and a number of other things. And then we're looking at the downstream effects of that in our functional assay. And then you can literally take assays like that and you read them off almost like, it's almost like a cartoon. That's more on the next slide. It's sort of like what it looks like, how many total transcripts. When you do these kinds of screens in here, what you see is that I got rich data on greater than 92% of all the cells that I'm measuring on. And if you ever do this thing at CRISPR-Cas9 at the single cell level, you know, nowhere near a 92% success rate on reading those cells. It's way, way down, as some of you perhaps know. Number of unique genes you're measuring per cell, and you know, another case where you get an incredibly rich set of information. Now with a readout like this, your controls are almost built in because you got enough plex, and I'm going to take you through one of these things because I think it's kind of interesting. I think we got enough time for this. Here's your guide RNAs. So let's take a look at slide number 5, where guide RNA number 5, and yeah, I don't know which one number 5, what the functional, what it's trying to do. Actually, I do know it knocks down some gene called KLK3, but anyway, so let's take a look at slide 5. That one's got guide RNAs in it. The one right next to it doesn't. And then I look at downstream signaling. Let's see how this guy reads out, actually. Let's maybe go. You can see in this particular case where we got, oh, okay, this is a case where it's a negative regulatory. Okay, so you knock something out up here, you actually turn on KLK3 or whatever. So you hit it here, shows up positive here, you got tons of it. The cell right next door that didn't have the guide hit it, doesn't have any of it there. And just convince yourself that's not a dead cell number 1, that just doesn't have anything in it. You can read over here, the housekeeper associated with cell 1 is still there. So here's the housekeepers going. There's the lack of a knockout, it's not there. Here's the guide where it is. There's the functional effect of that guide. So you get all this high density information, and it's all self-contained in a single experiment. All the controls, the functional readouts, all that stuff is controlled. So it's really a high efficiency way of getting information out on these types of screens. I blew up one just so that it's not so confusing to see so many things. But here's the detected guides, no guides over here. Where the guides, then where there's no guides, you see a whole bunch of this particular target. Where there is, you don't see any. And you get these pairs and triplets of information across all the cells that you're measuring at super high efficiency. Okay, that's sort of 2 very different applications. One's spatial biology. You're just trying to learn what Mother Nature is doing. The next one is like, I'm going to be active in what Mother Nature is doing, and I want to know all the phenotypic ramifications of that. Okay, now I'm going to end. I'm just going to show you a little movie of what it actually looks like, because you can't hardly appreciate this data until you sort of take a little spaceship ride and go right through all the RNAs. So here's a little section of tissue. The little gray, light gray things, those are DAPI nuclei. So we're looking at 1, 2, 3, 4, 5, 6, 7 or 8 cells on big blow up of a tissue region here. So you can actually see the individual RNAs. I'm measuring 1,013 plex. So I started to list the genes on the right-hand side, starting with the letter A. And, you know, that thing's going to travel a couple hundred yards in that direction with different gene names that I'm looking at. But, you know, those are just A through AG that I'm looking at, and there's a whole bunch of other ones in there. Now let's take a little look. So now you're actually sort of taking a ride, if you will. jumping into a single cell. You can see the RNAs in space. And you'll see as I get to sideways view, you can see about 7 to 10 planes of information where the RNAs are at above and below the nucleus. Now, it's hard to follow biology at 1013 plex, but I'm just going to put one little tiny biology story in here. Whoops, I was looking at the chat and stopped the movie there. One of the genes in this panel is something called MALAT1. MALAT1 is a long non-coding RNA that never makes its way out of the nucleus. Actually, I didn't know it was in the panel until we started looking at this thing. So keep your eye on the little orange ball, if you will. I'm getting rid of the other 1012, whatever, 1009 genes so you can actually see into something. So MALAT1 is a nuclear EEF1 and TMSB or whatever. Those are classic cytoplasmic RNAs. But now when we get on top of that single DAPI nucleus there, you can sort of see how the orange is right there centered on the nucleus, and EEF1 sort of forming a cone around the nucleus, if you will. Okay, you can sort of see it when I turn it all off. Let's bring them back in here. I think we'll bring out. Okay, there's MALAT1. Doesn't matter what cell you're looking at. See how they follow the nuclei, and then put them all back in there again. You can see most of them are not in the nucleus the way we made this panel, but it's easy to see things like that. Give you an idea of the spatial resolution that we're doing the RNAs at here, roughly. So you're actually looking at, somewhere between about 50 to 100 nanometers in X and Y for all these little RNAs. And then on the Z axis is not so defined. It's about probably 100, 200 nanometers. You end up with these really nice maps of these RNAs. I'm almost done. I think I'm down to about my last slide here. And then I know there's a bunch of things in the chat. We'll start picking up these questions. I want to end on the protein layer. So, when you're looking at RNA, you really don't see tissue morphology very well. That's just not, RNA is not good at that, but protein is great at doing that. So we're doing all that RNA work, and I highlighted the protein work for the cell segmentation. Now I'm putting a protein layer on top of that RNA slice where you can actually look and see clearly the tissue architecture that falls out of the RNA. That's a nice thing to be able to see with the protein layer. Here's a real simple protein one, so you can actually see with your eyes and sort of, convince yourself on how this readout chemistry works. If you just look at a couple of things at a time, one in the nucleus, one out in on the membrane, I can cycle at one place, and you can see those things over there. You can see where the little green signals are out on the membrane on that particular cycle. That's the CD45 thing we're lighting up. The histone H3 in red is on the inside. So there's a case like that. And then I can set it up as I cycle my reagents. I can spin that thing backwards. And now all of a sudden your red signal that was the green signal here now turns red because it's mapping the color doesn't map it's mapping a target, but how we cycle them and how we encode that information, each involves multiple colors. So it's not like one color stays in one place. They change patterns. The geography stays the same, but they change patterns in a known way. So that's why you see the green membrane thing goes to a red membrane thing on that next cycle, because that's the way we encoded the information. That means CD45. Similarly, the histone that started out red here, through this particular cycle, it's now green, and so on and so forth. That gives you an idea how it works, and that you can see with your eyes that it's sort of doing the right behavior. And when you get into real tissue with real plexes, of course, you can't just look at it with your eyes. You actually have to encode the information and decode. This gives you an idea how tissues look like. There's a cycle 1 of a tissue region, a blow up, cycle 2, 3, 4. Some of these cycles we color balance in particular ways, like cycle 4 is blue for a whole set of targets that we're sort of interested in. And you can sort of see how the information encodes in a spatial manner. It's the same kind of chemistry as RNA, but RNA just gives you a whole bunch of little punctate little RNA, single molecule RNA dots. When you do that with protein, of course, they group in a spatial way that's indicative of the protein-based geometry of the tissues. Okay, this is my last slide. So hopefully, I gave a little vignette on the thought processes and what it's like to do high-plex protein profiling, whole transcriptome profiling on what I call functional units of tissues. I think it's really powerful. And the fact is that, Mother Nature didn't make its little functional, the important functional areas in an XY coordinate grid that we can manufacture. You have to actually adapt to Mother Nature's way of organizing its functioning. And that's one of the powers of the mirror-based photocleavable oligo approach of the DSP is ideal for that kind of study. Because then I put all my whole transcriptome information, and if there's 2 million little glomeruli factories in a kidney, I can just focus my light exactly where those glomeruli are. And I'm not wasting my sequencing dollars doing all the stuff that's in between and all those other things. I'm getting whole transcriptomes exactly where I want them. And then, you get shapes that are really unusual, like you can see the collecting ducts. You need whole transcriptomes on those shapes. They're easy to get, so on and so forth. And I really do think you can recapitulate literally 50 years worth of research on a single pass of transcriptomes on a single tissue slice. Okay, that's not the whole picture. You got to go down into the private lives of these cells. And that's why we developed the Spatial Molecular Imager, where you actually go in there and get that detailed information. And you get how cells talk to each other and communicate. But you really want these two images to seamlessly go back and forth, how they're communicating, how they're organizing. And that's a lot of the work we're doing now is actually continue on the SMI. And now we're bridging the gap between SMI and the Genomics DSP to actually generate sort of a complete picture on how these tissues are actually organized and functions. So I think that takes us about right up to the hour, and I'll get off my screen shared. I see a number of things. I'll let Rong and Ahmet, do you want me to sort of take a perusal through the chat questions, or how would you like to do it?
Yeah, I think the technologies are unbelievable. Thanks so much, Joe. This is a terrific talk. I think I have lots of questions, but I'm probably gonna do that by email. But it seems like in the chat box already quite a few. You can read from bottom to up. I think several questions are kind of similar.
Okay, good.
Right?
Yeah.
Some technical questions in particular, how large the imaging area and the tissue thickness you can analyze using SMI, and yeah, maybe those questions first.
Okay, great. I see the one, how many proteins can you run? We know that we can just blast our way up into the thousands on RNAs. We actually don't know how many we're going to be able to blast our way up on proteins. We are targeting for the commercial launch to be doing roughly 100-plex protein panels on these kinds of tissue sections like this. But we're in new territory on some of these things. Some's new. The idea of encoding information on your proteins is actually pretty new, and I can space these things out, so I don't know what the ceiling is going to be on proteins. I got high hopes. I hope I'm doing this talk in 2 years, and we're doing 22,000 proteins in space instead of just like we are for RNA. We'll have to see whether we get there or not. The chemistry we're doing is identical. It wouldn't matter to me. The chemistry, I don't have to change the chemistry to do 10,000 proteins in space. It's the same thing I got for doing 10,000 RNAs in space. So I think the sky's the limit in terms of the chemistry and the hardware. Actually, we don't have to muck with the hardware either. So I'm hoping that we can just keep developing how to do these experiments. How big an area, what kind of spatial areas we're measuring. So that's an important thing, because as you guys know, cyclic high resolution work like this, we want to make a faster turnaround time associated with these things. So we're setting up the instrument actually you can put in I don't know, it's exactly 1 centimeter by 1 centimeter or so, but big pieces of tissue will be able to go into the SMI. Now you'll have to ask yourself, do I want to actually take that? And I really want all that detailed information on 1 centimeter by 1 centimeter. Sometimes the answer is yes, and then sometimes it won't be. So we're trying to set up the system so that you can do an overall scan of the whole thing, get a big picture, and then when you want to go in and look detailed on areas, that'll be set up for you to do it that way. Or you can take a smaller area and just do the whole thing in some kind of blind gridded manner. These runs will take, depending on your plex and how big an area, you're talking about running an experiment like this, in a day for smaller regions, say, whatever, let's say 20 square millimeters or something like that, or something like that in a day. You go to big, hundreds of square millimeters or even larger, they could be as long as 2 or 3 day runs. You'll be running 4 samples at a time, 4 individual flow cells at a time will be running as you run. So if you're doing a 3-day run, you got 4 whole sets of things that are getting done. If you're doing things where you cut your little tissues up, then each one of those things could have, let's say you got 10 or 20 in each one of those lanes, and you're doing that. So a 3-day run could be, you could have, say, 100 or 150 different tissue sections accomplished in a 3-day run at 1,000 plex, say. That's the sort of time frames we're talking about on these experiments. Okay, then protein at the same time along with this. Yeah, okay, so I answered the experimental runtime. We are working hard to make the RNA layer and protein layer come out on the same thing. All the panels at launch may not be combo on the same, but that's our goal, is to have everyone that way. So that's our goal. Okay, Z-section resolution. Again, those are probably on the order of about 200 nanometers or so on the Z. X and Y is much finer than that. Like I said, 50 to 100 nanometers in X and Y. Okay, I answered them. Actually, maximum thickness is really not a super maximal thickness, but we typically run on 5 to 10 micron sections typically. I'd love to talk about 3D versions of these things, but not quite there yet. Other than 3D within those kinds of tissue sections over a spatial domain of 10 microns or so. Okay, I got a question on pancreatic islets. It's one of my favorite ones. I was staring at full-blown, beautiful pancreatic islet data last night. Yeah, whoever is asking me, Andrea or whatever, send me a little note. I'd love to know what you're thinking about. I love the tissue substructures in a pancreas, and we're actually doing those measurements as we speak. Some beautiful data. Sometimes pancreas gets a bad rap because if you're chewing up the tissue, it eats up everything. All the Nc2 technologies, I mean, we have absolutely no trouble getting high resolution whole transcriptomes on pancreas and geomics or on the SMI, because we never chew the tissue up. That's the key. Just do it in situ, and I really like the stuff we're learning about the pancreas. Okay, targeted fish. I think we're actually getting pretty much through there. Probably could take some general purpose questions. I think we got through most of the little chat things wrong, and Ahmet.
Yeah, so that's great. So I have time to answer to ask my questions now. I think in your CRISPR experiment, were the samples like cells on a substrate in your flow cell? You're actually doing kind of in vivo tissue CRISPR experiment.
Well, eventually we'll get to there, Rong. I mean, I think I'm very interested in those, but we're starting out on known systems that are pretty well characterized. And it gives me a reminder to me, like that's a case where you're really just taking sort of a solution assay. Typically, they do these things with single cell and all this stuff you do this way. And we're saying, can we turbocharge that? And to give you an idea, like Matt Friedman, when we were getting done with these series of experiments and presenting this poster at AGBT, he told me that in our handful of experiments we did on the SMI, gave him more and better data than about 3.5 years of single cell CRISPR-Cas screening. And we got that in a handful of experiments. And I think there are going to be multiple assays like that. I'm really interested in doing tissues and all sorts of
Yeah.
organoids and all that kind of stuff, because then you'll get an extra layer of spatial heterogeneity on there.
Mhm.
So I'm very excited about.
Yeah, I think my colleagues here are doing sort of in vivo CRISPR mouse modeling. So I really think they should talk to you.
Okay, yeah, I'd be very interested. We're doing all of our probes right now, but actually, you know, the guide probes work like fine, but our phenotypic readouts are all human right now. We will be doing mouse for the end of the year, but yeah, send me something on. I'd love to see what's happening on the in vivo CRISPR-Cas9 in a mouse, Ron. That would be super.
Yeah.
Because
Yeah,
the heterogeneity there is amazing, actually, and to be able to, yeah, I love that idea.
Yeah, my second question is your video is really amazing. The last one, so it would be nice if we can make that like a VR, and maybe once I think things back to normal, you can talk to Disney whether or not they can do a
Take a little
kind of yeah, doing a ride, and then people really kind of do a kind of fun adventure into the cells, right?
Yeah, no,
cool.
real data, not like some science fiction thing. It's the real thing. I'm really hoping, I believe that what you're going to see, I mean, what I hope is that, you know, the biology's in there, right? So you got an RNA layer of information like that. You put a protein layer of information like that, and now you got multiple cells, you got cell-cell interactions. And I do believe you just dive in and let's see how all these things localize with each other and how they talk to each other. I think we're going to find some really interesting surprises down here. And then how they fit into the macro structures. Like we had that pancreas question there. You get these alpha and beta cells that are all stuck in the middle, and they're all intermingled, and they're doing these different functions. I really want to understand how that happens, and yeah, that's one of the things I'm looking at real closely right now.
So, quick question also from you. Great talk. Thank you, Joe. The amplification need for FFPE. Do you guys already incorporated in your probe design? Like, do you have a few?
Yeah, there are no other amplifications happening. It's all in that little 20 nanometer dot. Yeah, we when we were building SMI at the beginning, I actually added an extra channel way out in the red, thinking that may be the only way to get the other one. But we had enough signal that we can get it. Even in the, it's the FTSE channel that's the worst. Obviously, it's that green one, and yeah, we can measure over that.
Great. And for RNA protein multiplexing, I mean, my lab, we also do that actually, but the order seems to matter, right? Because they have different requirements.
Not here. Actually, this is the case. It's not cyclic imaging like that. All the business ends of the hybridization happen at once. So they all get equal footing in there. They all park in, which really makes a difference. Then that's just the it's a little tail readout is the thing you're doing, and they all happen at once. There isn't a cyclic biasing in terms of, because it is, if you're cycling whole targets, and as I change my biology, the second cycle of biology is different than the first cycle. So we tried to make all the biology one step. There is no cycling of the biology part. All that cycles is the little reporter part. And that helps a lot, Ahmet. So it's important to think when people are thinking cycling, you should always ask the question, you know, is the biology cycling? And if they say no, well, then that's better. If you actually have to cycle, a new batch of antibodies come in or a new batch of fish probes come in, that's where the problems come.
Right. And one also related question, not technology, but more biological question is, you talked about these unique T cells. So in immunology, you think that by using RNA-based classification versus protein, are we gonna soon redefine the T cell subtypes, or are we gonna revisit some of these definitions, or what do you think about this?
I think we're gonna go so much deeper in cell types, Ahmet. I feel no doubt about it. If you think about how all these cell types were determined in the first place, pretty much flow cytometry was where all these cell types came from. And they only have so many channels, and that's why we all track CD8s, and you know, CDs means surface. They're pretty much all surface antibodies. Why? Because there's flow cytometry that built that whole field. Now, when we go in from the RNA level, yeah, we're gonna see RNA-based subtyping, and that's gonna be big. It's gonna be much richer than the protein-based. No doubt about it.
Exciting.
Yeah. Also, it shows some correlation data with sort of Broad Institute cancer cell lines. Some of those are non-adherent cells, right? So that means actually your technology works very well for whatever cells, even just a kind of non-adherent single cells.
Yeah, I
people-
like going into that database. When you
Mm.
start going into mouse, it's kind of frustrating. You would think that there's so many great databases for mouse, but it's not like human, man. It's like I have to work harder in mouse to actually do my quantitative comparisons. But we're getting there. But it's some of the databases for human, all these cell line things are really rich.
Yeah, so just to kind of let the audience know, so it seems like the technology works well for, so it's not limited to adherent cells, right? So when you can kind of spin down the cells to a flow cell, it doesn't need to be adherent cells to.
Yep, and that's working also for geomics. We get some people now that are starting to think about taking CTCs. You put them into the geomics, and then you're doing whole transcriptomes on the CTCs and organoids and all these other things. So yeah, I'm pretty. And actually, I forgot to show my very final slide. It's if you want the SMI, that SMI instrument now is open up for what we call technology access programs. So just like in geomics, before we release an instrument, we got real world problems that come into the instruments before they go out. And we're at the stage now where SMI is taking them. It's got an email address on there. Maybe just find me. I'm pretty easy email to find. Wrong knows most of the people do. Just jbeechem@nanostring.com. Put SMI tap in your little subject line or something. I'll make sure and get it to the right person. If you're interested, if you got an interesting collaborative idea, we're looking at it now. If you got an interesting idea where you think it's something that you could actually be publishing on this year, and what you would love would be some of that kind of SMI kind of special sauce in your paper. Those are the kinds of projects we're looking for at this stage.
Yeah.
Great. Should we continue question and answer, or should we leave that for email exchange?
Can I ask one small question?
Absolutely.
Thanks for the great talk. It's a technical question again. So you said that your resolution is around 50 nanometers lateral resolution. Is it also true for the protein imaging?
It will be, yeah. But we'll have to see what the behavior. But yes, it will be the same.
But what kind of super resolution technique are you gonna use then?
Yeah,
you need a super resolution technique, right?
Yeah, that's right. Yeah, but we do pretty much most of our super resolution is 90% software. These are single molecules things that we're doing. So we can fit the tracks, fit the profile of the single molecules. It's 30 dyes, but they're locked in a 20 nanometer particle. So that's the super resolution angle we take. Does that make sense, Amy?
Yeah, I cannot get it, because when you have a really dense profile of the protein, it might be hard to get the super resolution without stochastic blinking, right?
No, it's a very good point. Because that's why I got a separate software group that work only on the protein quantification. Because it's really like you got it exactly right. RNAs show up like little punctates.
Yeah.
Proteins do not show up that way. And I call these things, I call it digital detection and analog detection of the proteins. Sometimes you don't care. When I'm doing some receptor that's got 20,000 in the membrane, do I really want to count that thing one at a time? Probably not. I call that's a great kind of a protein where you just do an analog readout of the geography. And you got some interesting transcription factor that maybe just a handful of them in there. Yeah, I really want to count that guy digitally. So our software groups are actually, it's a great point. Our software groups are working on multiple domains of quantitation, depending on the functional answer you want from your protein layer. And it's really a different set of questions that you want out of the protein layer than the RNA layer. And I'm super interested in finding people that are really as interested in the protein layer as I am. So everybody loves the RNA, but it sounds like you got some ideas. We got an active group on quantitating these things, and I'd love to hear ideas from outside people, actually.
Okay, thanks.
I had a question real quick. This is Mike Fenn. I'm from DuPont Therapeutics, if that's all right. Actually, you're talking about puncta, so that gets us obviously very excited with condensate field. We have a genomics system, which is fantastic, but so talking about the protein layer, we're very interested in being able to co-localize protein and RNA. What do you think about the compatibility of being able to use this system for screening, like cell-based type of screening? Do you think that's something possible?
I absolutely do. I actually think it can actually accelerate cell-based screening to a whole new level. Like you saw in that CRISPR-Cas9, you know, we did a couple of years worth of single cell CRISPR-Cas9, and we did it in 5 experiments. And we did it, all between all in a month or so for AGBT, and made the poster. And I really think imaginative cell-based assays can be turned way up. We have to rethink how we do them. And we're so used to just running solution-based versions of those. You got to dream up a sort of adherent version, and boy, that you can really accelerate. Yeah,
that's really exciting. I'm really happy to hear that. Thanks so much. This is fantastic.
Yeah, no, I recognize your name actually. That somehow you showed up on some screen I was at not too long ago. So yeah, send me a note. I'd love to talk with you.
I will. Thanks so much. Really appreciate it. Absolutely.
I think if you want to unmute yourself, maybe let's.
Oh, yep.
Okay. If not, I think there are questions, but please feel free to send an email to Joe for further details. It's almost 3:20, and we don't want to keep him, and he's very busy with creating all these innovative technologies. And I think with this, let's thank to the speaker again. I think I'm going to use this clapping for this great talk.
That's great. I love this format. I love the way you do this seminar series, because it actually allows us to just pretend like we're just sitting in a room having a science discussion. And I hope the rest of the people find it as enjoyable as I do. So yeah, send me a note. I love this stuff. And yeah, I'd answer. You send me a note. Connect on LinkedIn. Let's get connected. I think spatial biology people, I just hope we stay as connected as possible. There's just a handful of us that are really pushing the field forward. Let's stay connected.
Very exciting. And before we wrap up, I should also note that next week we have 10x Genomics, a complementary company. And from there, James Chell will be presenting Spatial Resolve, the whole transcriptome in FFPE samples with the 10x Visium assay. And with this, I think we're going to conclude today. And thank you again for participating. And thank you, Joe, again for this fantastic talk. And hope to see you guys next week.
Okay, good deal. And thanks for the invite. Appreciate it. Good luck. Good luck with your research.
Bye-bye, everyone.
Thank you, Joe, and thank you, everyone.